Fertilization and implantation. Fertilization is what? The fusion of male and female gametes. So where does this fertilization takes place? It takes place within the oviduct. So basically you can say that fertilization takes place in oviduct or fallopian tube. It takes place in oviduct and fallopian tube. Also in fallopian tube, more precisely, it takes place in ampullary region, right? कहाँ होते हैं ये? Ampullary region. So this is the ampulla where you have a egg, right? So what happened? The egg ovary releases egg and egg reaches to the ampulla of the fallopian tube, right? And from here, from the vagina, the sperm enters into the uterus. So now the sperm reaches to the fallopian tube, and then when it meet with egg, then what happened? The sperm releases acrosomal enzyme, right? The head of sperm releases acrosomal enzyme. This enzyme penetrate the wall of egg, that is zona pellucida layer of the egg, and then what happened? The head of the egg penetrated and the sperm releases its nuclei within the egg. All other part degenerated. Then again what happened? Here the fertilization takes place, the halted division started, and ultimately you will get a zygote, right? So after fertilization, what you will get? Zygote. Now let us see. So basically these are the you can say whatever I said, it is written here, okay? Once, just if you want you can note it down, because this is the notes only. Like, see, I'm just giving you all these notes, okay? So you can also, you will get a PDF, or you can make your notes directly from here. Once a sperm comes contact with the zona pellucida of ovum, right, and induces the change in the membrane that block the entry of additional sperm. Okay, so what happened? The sperm, once it comes and near the contact of it contact with the zona pellucida layer of the egg. They ensure monospermy and prevent polyspermy. So basically what happened now? Once the sperm fertilize with egg, suddenly what happened? A fertilization membrane formed around the egg. So due to that, what happened? Once the fertilization membrane formed around the egg, then due to this, guys, a fertilization membrane forms so that no other sperm can penetrate the egg, right? No other sperm can penetrate the egg. So you can say that ensure monospermy means only one sperm can fertilize the egg, not more than one, or you can say it prevent polyspermy. So only one sperm can fertilize the ovum. Now entry of sperm into ovum induces the ovum to complete its second meiotic division of secondary oocyte. So here what happened? The entry of sperm again started the secondary meiotic division. You will get ooted and second polar body, and then ultimately you will get zygote. So we have already discussed all those points. यहाँ तक कोई doubt? Any doubt? Anyone? Please ask me, guys, if you have any doubt. No? Right. Then what happened? What is cortical reactions actually? What do you mean by cortical reaction? See, immediately after the entry of sperm into the egg, a cortical reaction starts to check the entry of more sperm. Okay. In this reaction, what happened? The cortical granule present, like suppose this is the egg. So here you have a cortical granules present. These are what? These are the cortical granules, right? These are the cortical granules present beneath the egg membrane, plasma membrane, releases what? chemical substance between the ooplasma and the plasma membrane, that is called vitelline membrane. So plasma membrane of egg is called vitelline membrane. So what happened, this cortical granules releases a chemical, okay, once the fertilization done. These substance raises the vitelline membrane among the egg surface, elevated vitelline membrane called fertilization. Due to this chemical, they will form a vitelline membrane, which is only called as a fertilization membrane. The increased space between the ooplasm and the fertilization membrane and the chemical present check the entry of sperm, and due to which what happened, no other sperm can enter, because now this is the egg ooplasm, this is the vitelline membrane, so now the sperm are not able to penetrate them. If polyspermy occur, then more than one sperm enter the secondary oocyte, resulting cell has too much genetic material to develop normally, तो उससे abnormal पैदा होंगे. Then once the zygote form, right, once the zygote form, cleavage started in the zygote cell, right, the process started is cleavage. Okay, so what is cleavage? Basically cleavage क्या है? What is cleavage, guys? Cleavage is basically a repeated mitotic division of the zygote. Okay, it is what? It is a repeated mitotic division of zygote without growth, resulting a multicellular ball-like embryo called cleavage. So basically cleavage क्या है? Suppose you have a zygote, that zygote is a single cell. Now that single cell undergo a certain division, right? A single cell undergo a certain division, which is called a cleavage. Cleavage start soon after fertilization. So what happened then? Then this cleavage started just once the fertilization end, the cleavage started. Daughter cell produced during cleavage called blastomere. So now suppose this is the 2-cell stage, this is the 4-cell stage. After that again what happened? The 4-cell divided into 8-cell stage. Okay, 8-cell stage. So these cells are called what? These cells are actually called as blastomere. These cells are called as blastomere. The product of cleavage is called morula. Okay, so see, first of all, zygote, single cell, then 2-cell, 3-cell, 2-cell stage, 4-cell stage, 8-cell stage. So 8 to 18 cell stage is called morula. 8 to 18 cell stage is called morula. The morula continue, again these cells continue to divide and they form blastocyst, which is 16 to 32 cell stage. So 16 to 32 cell stage is called blastocyst. Alright, so the zygote undergo a series of mitotic cell division. Now, which cell division takes place here in the zygote? Mitosis, right, which is called stages. Now the blastomere, right, in the blastocyst. So basically you will get a blastocyst which is 16 to 32 cell stage, which is called blastomere, right. So the blastomere in the blastocyst arranged into an outer layer called trophoblast, and the inner is inner cell mass. So now suppose the blastocyst, the cells of the cell, or you can say the blastomere, arranged like this outside, right, and the few cell arranged like this. Okay, so this is called inner cell mass and this is called trophoblast. This is called trophoblast, right. So now the trophoblast, this trophoblast cells, the outer cells only, attached with the endometrium wall and develop placenta. So अभी ये जो blastocyst है ना, from the fallopian tube, now it enters into the uterus, and this trophoblast cells, which is the outer cell, now this trophoblast cell attached with the wall of the endometrium. After that, inner cell mass differentiated into embryo. So trophoblast cell form placenta, and inner cell mass ultimately develop into embryo. This inner cell mass ultimately developed into embryo, right? After attachment, the uterine cell divide rapidly and cover the blastocyst. So what happened? Once this attached, like suppose this is the uterus, okay, and the blastocyst attached with the uterus, then suddenly what happened? This endometrium wall cells started developing and they started covering, they started rapidly and cover the blastocyst. Blastocyst completely embedded in the uterine endometrium, and this is what we called implantation. So this is what we called implantation, when the blastocyst completely embedded into the uterine wall. Sex determination. Simply like you all know that about sex determination, like if XX is the female, XY is the male. In case of female, you have only XX chromosome. In sperm, carry X or Y chromosome, either X or either Y. So if X sperm meet with ova, female child. If Y sperm mix with the female, you will get the male child. Then we have pregnancy and the embryonic development, right? Pregnancy and the embryonic development. So after implantation, finger-like projection appear on the trophoblast called chorionic villi. So now suppose this is the trophoblast. Okay, so with the trophoblast, a finger-like projection form. Within the trophoblast, what happened? A finger-like projection comes out, right? And this finger-like projection, surrounded by the uterine blood vessels and the uterine wall. It is surrounded by the uterine blood vessels. So this chorionic villi. So now what is this? Now this is what? Chorionic villi, and this is what? This is your uterine tissue and blood vessel. Alright, so chorionic villi surrounds by uterine tissue and the maternal blood. Temporary association between the fetal tissue and the maternal tissue is called placenta. So now the association between the uterine and the uterine tissue and the placental tissue, basically trophoblast tissue basically forms the placenta. So chorionic villi and the uterine tissue forms the placenta. Placenta, the function of placenta is what? The embryo is now connected to the placenta by umbilical cord, which transports substance to and from the embryo. So now this embryo is connected with the placenta with the umbilical cord. तो अभी जो embryo है ना, वो placenta से नहीं, because embryo formed from inner cell mass and trophoblast form placenta. So the embryo connected with that placenta with the help of umbilical cord. This facilitates the transport of oxygen and nutrient from mother to embryo. Remove carbon dioxide and waste material from the embryo. Act as a endocrine gland and produces several hormones like human chorionic gonadotropin, human placental lactogen, estrogen, progesterone, relaxin, and pregnancy. So these are the hormones which is releases from the placenta. Now embryonic development. Okay, embryonic development. What is embryonic development? After implantation, the inner cell mass of blastocyst differentiate into an outer layer called ectoderm and endoderm. So once the implantation done, so again the inner cell mass which develops into the embryo, the outer layer of the inner cell mass form ectoderm, the inner cell mass form endoderm, and the middle one form mesoderm. So basically these are the three germ layer from which the whole body organ form. The inner cell mass called stem cells. So whatever the cells we have in the inner cell mass na, they are called as a stem cells, because these cells are capable of forming these cells are only capable of forming what you can say that these cells are capable of forming all the types of cells we have in our body. Okay, so this is what we called stem cells. Parturition and lactation. So parturition, guys, the period of pregnancy is called gestation period, right? So here the period of pregnancy, basically 9 month, the period of pregnancy is called as the gestation period. Okay, ejection or expulsion of delivery of fetus is called parturition. So basically like fetus took 9 months to develop. After the development, when the fetus releases from the body, that is what we called the parturition. So the release or the expulsion of fetus from the body is called parturition. Parturition basically occurs due to the contraction of myometrium wall of uterus. So whenever there is a contraction in the myometrium wall of a uterus, then only the develop. The signal of parturition is originated from the fully developed fetus and the placenta, called fetal ejection reflex. So the signal of parturition, basically the signal of parturition originated, कहाँ से ये सिग्नल आता है कि अब मुझे बाहर निकलना है? Who will give the signal? The signal given by the fetus itself and the placenta. So the fetus and the placenta both will provide the signal. Fetal ejection reflex trigger the release of oxytocin from pituitary. Alright, so oxytocin again induces contraction in the endometrium, stimulate expulsion. After delivery, the placenta is also expelled out of the यूट्रस. सो डिलीवरी के बाद जो प्लेसेंटा है वो भी यूट्रस से निकल जाता है. राइट, नाउ लेट्स हैव अ lactation. Lactation, basically the mammary gland of the female more differentiated during pregnancy. So we have already done with the mammary glands. Mammary glands produces milk. Process of milk production after the delivery is called lactation. In the first few days, whatever the milk produces by the females or milk produces by the mother is basically called as colostrum. Right, in the first few days, whatever the milk produces. Colostrum contains several antibodies. So these colostrum contain several antibodies which provide immunity to the newborn baby. Alright, so this is what we call lactation. So these are basically the topic of human reproduction. You are done with the human reproduction. Majorly the question you will get it from the gametogenesis. You should know the spermatogenesis, oogenesis, their location, what type of cell division takes place, where meiosis takes place, at what stage mitosis takes place, at what stage what will be the number of chromosome, when it is diploid, when it is haploid. So these are the questions you have to prepare. Right, after that gametogenesis, menstrual cycle is very important. menstrual flow, from the flow of menstrual cycle you will get a question, and after that you will get a questions from something from placenta, right? How the placenta form and the hormones coming out from placenta. And lactation parturitions are not very much in detail you have to learn. You have to learn in just the elementary ideas. You should know the elementary idea about these parturition and the lactation.